All right, in this section, we're going to look at some things we probably haven't seen a whole lot of before. So we're going to start talking about cells. Originally in 1665, there was a man named Hooke who examined a piece of cork under a really crude old school type microscope, and he identified these little rooms in there because they thought they were empty and they kind of looked like monks' kind of cells. So he called them cells. So Leeuwenhoek, working at about the same time, used more refined lenses to describe living cells from blood, sperm, and pond water. So since the days of Hooke and Leeuwenhoek, we have improved microscopes and have vastly expanded our view of the cell. This is a lot of cool stuff that we found over time. Just the intro. I mean, we can see this kind of stuff now, which is really difficult to see and very, very cool. So we're going to talk about a lot of these parts coming up in the next lecture. So we will introduce the cell, and honestly, we could do some of these other ones, but they're going to name specific parts and their functions. So really in this lecture, we may just introduce the cell. It'll be about as far as we can make it. So we need an essential question for this. I think our essential question should be, what are cells? That's what we're trying to figure out. And how are they classified? Because there's a couple of different types of cells, and so we want to be able to distinguish between them. So what are cells and how are they classified? So a variety of microscopes have been developed for a clearer view of cells and cellular structure. A lot of cool microscopes. We'll look at some of them. But the very first microscopes were light microscopes. That's what we call them. Because visible light passes through a specimen, then through a glass lens, and finally projected into the viewer's eye. So hopefully we'll be able to get one of these. If not, I do have a very old school version. I have a light microscope that doesn't use like a little lamp. It uses a mirror from the 1930s. It's a beautiful thing, so we're going to look at it. But what we do with these is we're going to magnify things up to maybe 1,000 times. Some fancy ones can do a little more, but we're not that great. Okay. So magnification, think about we're going to magnify something. This is an increase in the object's image size compared to its actual size. So we just make it look bigger. Okay, so that's that. Resolution is a measure of the clarity of an image. In other words, it's the ability of an instrument to show two nearby objects as separate. So resolution is more about clarity. So magnification is bigger, resolution is clarity, or kind of, you might think like focus as well. Okay. Also, you get better resolution, kind of, you could think computer terms when they've got more pixels, you can see better resolution, that kind of thing. So microscopes do, in fact, have limitations. The human eye and the microscope have limits of resolution, so the ability to distinguish between small structures. My human eyes, in particular, have really some limits in resolution. If you have some eye issues, sometimes that happens, but that's okay. We still get the job done. So therefore, the light microscope can't provide details of the small cell structure, and we'll look at those. We can only see so far, really is what that's saying. So using light microscopes, we've studied microorganisms. So micro means small, so we're little organisms. We've also looked at animals and plant cells. And we looked at some structures within cells. Not a whole lot, but there'll be a few that we can see. In the 1800s, studies were led to the cell theory. There's actually another part to this, but these are the main ones, is that all living things are composed of cells, and all cells come from other cells. We might try to investigate the third part of this later, but at very least, we do need to know these two parts of the cell theory. So, looking at a little bit of magnification and looking at some resolution and different things. This is a paramecium. We'll talk about him later. He's a little protist guy. He's a cutie. So that's one type of microscope we can see him as. That's another one. Same guy, just different kind of microscope to look at it. Same guy as well, but now we're looking at the insides. and same little guy, a different microscope. So now that we've got more advanced technologies, we can see them much differently. Okay, so with your human eye, you can see obviously human height. This is kind of a scale. I have a really, really cool scale for us to look at on this one. But human height, we can see chicken eggs, we can see frog eggs, we could see the paramecium, that little guy we just looked at. You could see him with your naked eye, but it's a struggle. Human eggs, so under the light microscope, we can definitely see paramecium, and we can probably see down to the mitochondria, but it's going to be tough. We'll definitely be able to spot some bacteria if we stain them properly, which is a lot of these guys is we have to kind of stain them so we get a little bit called contrast. But that happens. And then we have these cool things called electron microscopes that allow us to see all the way down to the proteins and lipids, which are really, really cool. We can't see much smaller than that, but we at least kind of start to see some of those macromolecules. So in the beginning of the 50s, scientists started using a very powerful microscope called the electron microscope. And there really aren't that many of them around. I don't think that they're all that accessible. I do know of a group that you could sort of almost hijack their microscope. They'll let you kind of control it remotely. But I don't know how far in advance they want that scheduled. So I haven't managed to get it figured out yet. But maybe if I can find a day later on, we can see if we can schedule it during like a weird time of the year, something like that. But it's such a cool thing, electron microscopes. These view the ultrastructure of cells, so kind of the really small, unique things. So instead of light, an electron microscope uses a beam of electrons, so it just kind of shoots little electrons out there, and we'll kind of get an image from that. The electron microscopes resolve biological structures as small as 2 nanometers. That's 2 times 10 to the negative 9. Okay, and it can magnify up to 100,000 times. So we can see some small things, real small. There are 2 types. One is a scanning electron microscope, and those study the detailed structures on the surfaces because we're scanning the outside of it. So we're just like a scanner. We're just looking at the outside. Okay, so, but there's this cool thing we also have, which is a little bit newer, is the transmission electron microscope. This gives us details of internal structures, so we can see what's going on the inside. But if we didn't use differential interference light microscopes, amplify details and intensity, so the structure of the living cell appear almost 3D. So that was kind of that last one. We do have some interesting ones that will put the images together. I had some friends who worked with, small little mites, and so they would take kind of pictures at different places, and they could compile all the clarity together, and you get a really cool image. So that's what these guys are doing, is trying to kind of take several pictures and put them together so you get a nice, clear 3D image. It's really neat. Okay, cell size. So we'll do a little bit of a demo with this one, but cell size has to be large enough to house DNA, proteins, and other structures that we need to survive, but also be small enough to allow for this surface area to volume ratio so we can exchange with the environment. We want to be able to get things, so if we have a cell, we want to be able to get things in that we need and take things out that we don't need, some waste, as quickly as possible. So this is what the surface area to volume ratio is about. Okay, so if we kind of look here, we could do the surface area of this particular cube, and if it's 3 whatever units they are, maybe it's centimeters for this case, so we use 3 centimeters, the surface area comes out to 54 units, and the volume, when we do 3 times 3 times 3, is 27. So you do surface area to volume ratio, and you come up with 2. Now if we take one little cube out of this, that's a 1 by 1 cube, notice what kind of, and we're actually, I guess, using the whole thing. So the total volume is still the same. We still have kind of 3 whatever units across. So the volume still stays the same, but when we've kind of added these little, I don't know, nooks in here, it increases our surface area a whole lot. So if we were able to, if we're trying to get, let's say, water in, notice it would take a long time before we got to the center. But if we could kind of move in this way, we could get to the center a lot quicker on all of these. Okay, so it just makes things a lot quicker to have more surface area per volume. We'll demonstrate that a little bit differently a little later on. Okay, this is probably a good place to take a break because we'll go into kind of the membrane. So if you need a break, which I always recommend, pause the video, maybe look at your notes a little bit. Can you chunk them, write some questions, start a summary? Do that. Pause the video and then come back. Okay, so the plasma membrane, okay, is a flexible boundary between living cell and its surroundings. Yay! Okay, so this is going to kind of provide a barrier, a boundary. It's going to be selective. What's included here, something we learned about in the last unit, are phospholipids, and they form a two-layer sheet called a phospholipid bilayer, in which hydrophilic heads face outward, and they're hydrophilic because they like water, so they get along with water. Okay. So they're a little bit polar on the outside, and they're exposed to water. Then we have hydrophobic tails, so these guys do not get along with water. Okay. And they're going to pour inwards towards each other and be shielded from that water. Okay. So what that looks like, oh, let's see. We'll draw it out here for you. What it'll look like is, so here are kind of our heads, as they look a little bit like this. And what will happen is that these hydrophobic tails will point in towards each other. There'll be a much better picture later, but that's what they're going to look like. Well, it's a little off, but hydrophobic heads right here, or hydrophilic rather, heads, and then hydrophobic tails, notice are in this kind of layer in the middle here. Okay, so the membrane proteins are embedded in the lipid bilayer. So the thing I just kind of made for you look like jellyfish waving at each other. Okay, sometimes there'll be proteins in there, and so they'll look sort of like that within this little layer of jellyfish waving at each other. Okay, quickly. Some proteins form channels, so sometimes you have like a tube that shield ions and other hydrophilic molecules as they pass through that hydrophobic center. Other proteins serve as pumps, so they'll use energy to actively transport molecules into and out of the cell. That's a little weirder one to draw, but sometimes they're pumps, so they'll take a little energy in order to get it going. So imagine that's like a little burst of energy. It's needed here to make this thing go. Okay. So here's sort of what it would look like. Much better diagram than what I'm drawing, but maybe you can see it. Notice the hydrophilic heads on the outside, those little gray parts. The hydrophobic tails are pointed inwards towards each other. This box indicates what one little phospholipid looks like. So it's kind of the one head with the two little jellyfish-looking legs coming off of it. That's each a phospholipid. For each of these particular proteins, it says this part would be the hydrophobic region because it's getting along with these tails. And then this part up here would be like a hydrophilic region because it would get along with the water on the outside. Okay, so that's that part. Notice there's a channel, sort of like what I was drawing, so something can move through the channel that's kind of selected for here. So those are some of those things. Bacteria and archaea, so we're kind of changing route here. We got a little bit on the membrane. We'll go into more detail on that guy a little bit later through cells. But this part we'll talk about actually get the types. So this is going to be kind of looking at those types again. So bacteria and archaea are prokaryotic cells. All other forms of life are eukaryotic. So eukaryotic cells are distinguished by having membrane-enclosed nucleus and membrane-enclosed organelles that perform specific functions. Prokaryotic cells are smaller and simpler in structure because they don't have a nucleus and they don't have those little organelles. So they're kind of going to be small and sort of wimpy looking. They're wimpy looking, but some real nasty stuff ends up being prokaryotes, like strep throat or MRSA. There's some like pretty wicked diseases that these, little simple cells can mess you up. Okay, so prokaryotic and eukaryotic cells do have some things in common. So this is going to be kind of some commonalities between the two of them. Some things that they're all going to have. No matter what cell they are, they have all of these things. Okay, one of is the plasma membrane. Doesn't matter what kind, doesn't matter where it comes from, they all have that membrane. They all have an interior filled with a thick jelly-like fluid called cytosol. The cytosol is like I call it the juicy stuff in the middle of the cell. Juicy jelly stuff. Okay? They're also going to have chromosomes, which carry the genes made of DNA, so they all have to have some kind of genetic material, every cell. They also have to have ribosomes, so tiny structures that make proteins according to the instructions of genes. So, although not added here, we could make the note that also prokaryotic and eukaryotic cells have to have proteins. I mean, what's the point of having a thing that make proteins if we don't actually keep the proteins and use them? So that's another thing that all cells will have as proteins. Okay. Inside both types of cells, there's cytoplasm. However, the eukaryotic cell, this term only refers to the region between the nucleus and the plasma membrane. Just so you get an idea, honestly, cytosol and cytoplasm are, one of them is like it refers to a region, the other one's the juicy stuff, but I use them, I'll admit, I use them interchangeably. If you want to look it up and be more correct and then notify me of my correctness or incorrectness, have at it. I'm happy to be told what to do correctly on that one, but I will use them interchangeably just because that's how I learned them. The distinctions sometimes happen later on, so if you figure the distinction out, you can tell me. Okay, in a prokaryotic cell, we have the DNA coiled into a region called the nucleoid region, or nucleoid space. It's just kind of like a nucleus-like space. Not really a defined space, but it's just kind of like a general area. And there's no membrane to surround that DNA. So here's maybe a cell, and instead of having kind of, here we'll go over here, what we think of as like a little nucleus with a little membrane around it, they just kind of have their genetic material in a space. It's not all over the whole cell, but it doesn't necessarily have a nucleus to surround it. It's just in a region. So that would be the nucleoid space. Outside of the plasma membrane of most prokaryotes is a fairly rigid, chemically complex cell wall. It'll actually help protect the cell and also maintain its shape. So they've got, there'll be some pretty cool things there. Some prokaryotes have surface projections, so little things that kind of like poke out of it. I don't know, almost like hairs, they come out. Short projections help them attach to each other or their substrate. So sometimes you'll see like small things that come off of it. Longer projections are called flagella. One by itself is a flagellum, and they'll propel the prokaryote through a liquid environment. They're long and maybe squiggly. And they're for kind of whipping around and making sure that this thing goes this way. Okay, so that's a flagella. The flagellum, excuse me. Okay, so here's kind of what we see. These are the shorter projections I was talking about. They're called fimbriae. We might have some ribosomes in here, as we talked about. Here's that nucleoid space, the general region where we would find the genetic material. Our plasma membrane is kind of this like maybe purpley looking thing inside of it. It's hard to see, but it's in there. Then we have a cell wall on the outside, which is definitely going to be the yellow part. Oh, it's doing this one. There's also, you can have a capsule on the outside. So purple is plasma membrane. I suppose the orange is actually cell wall. And then for this one, the yellow is the capsule. My bad. Okay. Bacterial chromosome is what's on the inside, and this one's called rod-shaped one. You notice on the outside there's 3 flagella, and they're for getting this thing to move around. So this is E. coli, Escherichia coli, and that sort of colored TEM or transmission electron micrograph of what this guy looks like. So you start to see this general kind of characteristic. The membrane is like there, capsule right there, flagella. You can kind of see those. All right, eukaryotic cells contains a bunch of other stuff, and we're going to take lots of time in the next lecture to go through all of the organelles. It's painful, but we're going to do it because we can handle it. Okay, so a eukaryotic cell contains a membrane enclosed nucleus. It kind of, or a little bit reminds me of like the egg yolk. It's in the center. And then we have various organelles, which literally means little organs. And they're just kind of like organs, and they'll perform specific functions. They'll do certain things in the cell. You know, there it is. So there'll be more of those, but they're little organs that do certain things. The structures and organelles of eukaryotic cells perform 4 basic functions. And actually, when we kind of go through them, we're going to go by these functions. So this will be kind of where we stop things, but it's good to start with these functions. First, we've got the nucleus and the ribosomes, and they're involved with genetic control of the cell. So we'll talk about those guys first. It's always based kind of on function. So nucleus and ribosomes. Then we have kind of a section that are endoplasmic reticulum, Golgi, lysosomes, vacuoles, peroxisomes, and they're going to be involved in the manufacture, distribution, and breakdown of molecules. It'll also be helpful because we really will go through every one of these guys and their functions next time. So if it helps, you can kind of get started with writing out some of these guys. It may save you some time because I tell you that will not be a short lecture. I'm sorry. But if you look through some or you look through part of your book, it'll help you kind of get started on those. Then we have kind of the energy processing ones, and those are going to be mitochondria and chloroplasts. Okay, those are those two. And then involved in structural support and movement and communication, we'll have the cytoskeleton, plasma membrane, and cell wall. And you'll notice that with like cytoplasm, cytosol, cytoskeleton, when you see cyto, really on kind of any part, it's going to refer to just a cell. So this is the cell skeleton. But those will be kind of what are involved. So those we can all look forward to talking about in greater detail in the next lecture. The internal membranes of eukaryotic cells partition it into compartments. Then we have cellular metabolism, and that's all the chemical activities of cells with their various organelles, kind of all that work will occur within those organelles. Almost all of the organelles and other structures of animal cells are present in plant cells, so we're going to see a lot of similarities. We'll talk about a few differences, which are worth noting as we move kind of towards that section. So the exception is that lysosomes and centrosomes containing centrioles are not found in plant cells. So these will be animal only. Okay. And only the sperm cells of a few plant cells actually have flagella. Okay. So these are animal only. Plant cells are cool enough to have some things that animal cells do not. Okay, so these are also important to know, is that plant cells have a rigid cell wall that contains cellulose. Notice it's that sugar we talked about, OSE. They've got these cool things called plasmodesmata, and because they have this rigid cell wall on the outside, they really need to have little holes poked in it, so that way they can connect with other cells. So imagine you've got these thick walls, and they're all together. Well, it's like a fortress on a fortress. How are they supposed to connect? So there'll be kind of little holes within them, and those are called plasmodesmata. So the little channels, little holes poked in the cell wall so the cells can talk to each other, because they got to do that. Chloroplasts are another one. This is where photosynthesis will occur. And they have this cool thing called a central vacuole, which stores water and a variety of other chemicals. So all of those things in plants. All right, So an animal cell, we can see some of these, which we'll talk about later. In the nucleus, we've got cytoskeleton with its little tubules, peroxisome, plasma membrane out here, a pair of centrioles is the mitochondria, lysosomes, which are animal only, the Golgi apparatus, smooth and rough endoplasmic reticulums. Okay, or maybe reticula. I don't know. Okay. Again, this is a plant cell, so I'm not sure I made that very specific in the other one. So, by the way, this is an animal. Oh, okay, terrible pen writing there, but that's an animal. So this one's obviously a plant. You can see this nice rigid cell wall, which is pointing to kind of right here. And you can see these little holes poked in there, so that way those different cells can talk to each other. That would be the plasmodesmata. You can see that here too. Okay. But notice some of the same stuff. There's Golgi. Look at the peroxisomes. I was thinking lysosomes. Peroxisomes are over here. Plasma membrane is still kind of on the inside. Cytoskeleton again, mitochondria, smooth and rough endoplasmic reticula. But then we've got the different central vacuole. So really our kind of different guys are over here. Central vacuole, chloroplast, cell wall. Plasmodesmata. Okay? So that's enough stuff, but notice we're going to go each section by each section. So if you would like to save some time on that one, you can kind of get started in thinking about it, because that's the big thing that we'll do in the next lecture, is go through all the organelles and their functions. So if you start a little ahead of time, it might save you some time in the future. Okay, so with that, let me know, email me if you have any questions, and we'll just keep moving along.